So I will be speaking a bit about pathogenesis, but I'm going to primarily talk about vaccines. The COVID-19 pandemic, I think, truly requires no introduction because of how it's profoundly affected all of our lives. So why do we need a COVID-19 vaccine? And principally because it is likely required to end this pandemic, and it also could be important to control future outbreaks of this or related viruses. And regardless of the clinical relevance of a vaccine, then vaccine research is an important contribution to science because our understanding of immunology and immune correlates of protection are really central for viral pathogenesis. So we started out early this year with trying to address two critical questions for COVID-19 vaccine development. These first two papers really were pathogenesis papers, and we felt that an understanding of protective immunity from a pathogenesis perspective would really be critical for not just our vaccine development program, but really all the vaccine development programs. So the two questions that we addressed first, is there natural protective immunity? Will individuals who recover from COVID-19 be protected against re-exposure? And second, is there vaccine-induced immunity? And if so, what are the immune correlates of protection? These are questions that are critical to address in humans and will require large-scale human studies that are actually now ongoing. But to gain early insight into these questions of immunity and pathogenesis, we performed studies in the rhesus macaque model. So in the first study, and these two were published back in May, so I'll go over them quite quickly. In the first study, we really set up a rhesus macaque model that can be used to study pathogenesis as well as vaccines and therapeutics. In this model, we infected 9 rhesus macaques with a challenge stock of SARS-CoV-2 at 3 different doses, 10 to the 6, 10 to the 5, and 10 to the 4 PFU, as in group 1, group 2, and group 3. We showed that there was good infection at all 3 of these doses. And here we look at viral loads in bronchoalveolar lavage. And essentially at all doses, we essentially get 7 to 14 days of virus replication in rhesus macaques in the lung. In nasal swabs, there's even more prolonged virus replication at all 3 doses. Viral loads peak at around day 2 and then decline over time. And there's virus replication observed in nasal swabs for approximately 14 to 28 days. We also looked at the histopathology of the viral pneumonia in rhesus macaques. And this is really similar to what several other groups have seen in non-human primates. Overall, monkeys don't get very sick. We published a hamster paper a few weeks ago that do get very sick. And there, these findings are even more pronounced. So in macaques, the pneumonia is oligofocal. It's not encompassing the entire lungs, but it's patchy. But in the areas that are affected, there is really substantial viral pneumonia. Panels A and B show acute inflammation in the lung. Panels C and D show a terminal bronchiole with sloughed epithelial cells. In panel E, we show a multinucleated giant cell. In panel F, we show the formation of hyaline membranes. In panels G and H, we show immunohistochemistry for the SARS nucleocapsid, showing infection of a multinucleated cell in G and a pneumocyte in H. This is done with Manny Martineau at Tufts. In panel I, we also looked at RNA scope analysis with Jake Estes at OHSU, and we see clear virally infected cells in areas of acute inflammation. In panels J, K, and L, we look at a large bronchus, and we see in J by H&E staining acute inflammation and necrotic nuclei migrating to the surface. In panels K and L, we see by RNA scope, as well as in L by immunohistochemistry, direct infection of ciliated bronchial epithelial cells, all hallmarks of viral pneumonia. And in this paper, we did a study of natural immunity. We allowed these 9 animals, we allowed them to recover, and the viral loads in bronchoalveolar lavage are shown on the y-axis. After the primary challenge, these animals recovered by about day 21. And then on day 35, we re-challenged them with a similar dose of SARS-CoV-2, and there was substantial protection against rechallenge. We also showed that these animals developed both humoral and cellular immune responses. And these show some early proof of concept data that natural immunity, at least in this particular model in macaques, gives substantial protection against rechallenge. This doesn't say anything about humans. This doesn't say anything about the durability of this protection, but it does show proof of concept that there is indeed natural protective immunity to this virus in this model. We also performed an early vaccine study with DNA vaccines to get an early sense as to what protective efficacy might look like with more potent vaccines. These DNA vaccines are not vaccines that are proposed or are being developed clinically. These are purely research tools to learn about antigens and immune correlates of protection. And because we could make the DNA vaccines very quickly. So our first vaccine study, the main outcome is shown here. We had a group of 10 sham controls and then various smaller groups of vaccinated animals that received different spike protein immunogens. In the sham control group, all the animals were infected, and here we show viral loads in bronchoalveolar lavage. Peak viral loads in the sham control group were about 5.5 logs. And this was reduced by about 3 to 4 logs in the optimally vaccinated groups. One finding we were able to discern in this study was an early immune correlates analysis, because some of these vaccines showed considerable efficacy, and some of these vaccines that we tested early on actually did not show very good efficacy. And so the variation in outcome allowed us to look at an early immune correlates analysis, and looking at a variety of different immune parameters, the best correlates were the neutralizing antibody titer induced by the vaccine. So here we show a clear correlation, a clear inverse correlation between the neutralizing antibody titer induced by the vaccine and the degree of protection in the lungs. So moving now towards the vaccine program that we are developing clinically, it's based on an Ad26-based vector, which is a replication-incompetent common cold virus that's been developed previously together with Johnson & Johnson for multiple vaccines, including HIV, Zika, Ebola, and RSV. This vaccine platform has been given to over 100,000 people to date, and regulatory approval has been obtained for the first Ad26 vaccine for Ebola in Europe. There's also been pediatric experience with Ad26 RSV vaccines down to 12 months of age, because obviously rapid age de-escalation into pediatric cohorts will be important for protecting school children. So we started working on the Ad26-based COVID vaccine in early January when the virus sequence was released. By January 13th, we had a series of synthetic genes that we ordered to start the vaccine design efforts. By the end of January, we partnered again with J&J, with whom we worked for our Ad26 HIV and Zika vaccines. In February, we immunized mice and rhesus monkeys. By the end of February, we had a challenge stock. And in early March, we started to develop both small and large animal models of disease. We've largely settled on hamsters as our small model of disease and macaques as our large model of disease. In May, as I mentioned, we had our initial studies on natural and vaccine immunity. And about 6 weeks ago, we published the protective efficacy of the Ad26 platform in non-human primates. In late July, we started phase 1 and 2 clinical trials with this vaccine. And if all goes well, then we hope to start a phase 3 efficacy trial this month. In terms of the landscape for vaccine trials, of course, this is one of multiple programs that are either in or soon to be in efficacy trials, including the first vaccines to be tested, the RNA vaccines in the US, the first vaccines to be tested, the RNA vaccines of Moderna and Pfizer, the ChAd-Ox vaccine of AstraZeneca, the Ad26 vaccine, which I'll tell you about, and then protein vaccines from Novavax and Sanofi. And I'll just, I'd like to give credit to the previous speaker, Jason McClellan, who described the proline stabilization mutations that have actually been included in the majority of these vaccines, including in our vaccine that we are moving forward. So I think Jason and his colleagues deserve a lot of credit for that work. So we published the initial protection observations with a single-shot Ad26 vaccine about a month ago. In this study, together with our partners at J&J, we produced 7 different Ad26 vectors expressing different forms of the spike protein, including spike proteins that have either the wild-type leader sequence shown as the black triangles or the TPA leader sequence shown in red, full-length spike proteins or deletions at the cytoplasmic tail or the full transmembrane region, as well as the native sequence, and really as a head-to-head comparison, the mutated versions with the furin cleavage site knocked out and the 2 prolines. We weren't, the Hexapro was not available when we initially designed these, but we did incorporate several constructs that included the 2 prolines. And I'll just say that the vaccine construct that worked the best is the one at the bottom, which we called S.pp. Essentially, it's the wild-type leader sequence, the full-length spike, and then the mutations, which include the furin cleavage site mutation and the 2p stabilizing mutations. The study that we did included 52 rhesus macaques that were on the older side, 10 to 12 years old, mixed gender. Because of the potency of this particular platform in generating responses after a single vaccination, we decided to look at the immunogenicity after a single shot vaccine, and then decided to proceed with a challenge to see whether a single shot vaccine would be sufficient for protection. We used a dose of 10 to the 11th viral particles of Ad26 vectors, expressing one of 7 of these spike variants. There were 32 vaccinated animals, 4 to 6 per group, and we had a large control group of 20 to look at the consistency of viral infection, essentially as a pathogenesis question in this model, to see whether we would get truly consistent protection in a large number of animals, and then to also have substantial power to compare with the vaccines. After this single vaccination at week 0, at week 6 we challenged these animals with 10 to the 5 TCID50 of SARS-CoV-2 by the intranasal and intratracheal routes. These are antibody responses induced in these animals. These are binding antibodies by an RBD ELISA, and you can see that all of these vaccines generated binding antibodies by week 4. The initial TPAS construct had the lowest binding antibodies. These are neutralizing antibodies by a luciferase-based neutralization assay, a pseudovirus neutralization assay, and all of these animals developed neutralizing titers after immunization by week 4. The lowest neutralization titers were in the TPAS group again, and the highest neutralization titers were in the S.pp group, shown here, several fold higher than the other groups. These data suggest that the wild-type leader sequence is better than the TPA leader sequence, at least in this particular platform. The full-length spike is preferable to a deletion or a soluble fragment, and the 2 proline stabilized mutations appear to have increased immunogenicity. These pseudovirus neutralization titers are about 4-fold higher than titers that we observed in convalescent monkeys. These are actually the monkeys that were protected against rechallenge, so about 4-fold higher than titers that we know were protective in monkeys, as well as about 4-fold higher than a cohort of convalescent humans from the Brigham and Women's Hospital in Boston. Together with Galit Alter, we also looked at antibody profiles and function, and these, of course, are complex antibody profiles, not just neutralizing, but they have multiple different subclasses and FCR binding attributes shown in red, and multiple different functions shown in blue, in addition to neutralization. And again, the SPP vaccine generated the broadest and most comprehensive antibody profiles. The vaccines also generated T-cell responses. These are interferon gamma and IL-4 ELISpot responses that are negative in the sham control group, and in the S.pp group, the animals generated, I would call them moderate magnitude interferon gamma responses, and really no IL-4 responses, indicative of a Th1 response, which is as we would expect from this platform. We next looked at protection and following challenge. These are viral loads in bronchoalveolar lavage using a subgenomic RNA assay that is relatively specific for replicating virus. In the sham control group shown on the left, 20 out of 20 sham controls were infected with peak viral loads on day 2 following challenge between about 4 and about 6 logs of virus. The median bronchoalveolar lavage subgenomic RNA level was about 4.5 to 5 logs. Viral replication was also sustained for a number of days and generally resolved by approximately day 10. In contrast, in the SPP vaccinated group, there was no virus detected by subgenomic RNA PCR in bronchoalveolar lavage in any animals at any time point, indicative of complete protection in the lungs. If we look at the other 6 vaccines, all of these other vaccines also showed a degree of protection. Between 0 and 2 animals showed breakthrough virus in each of these vaccines, and the viral blips were generally lower in magnitude and shorter in duration than in the control group. So essentially all of these vaccines generated some degree of protection in the lungs with the SPP vaccine, as well as the TPA vaccine being the best. If we look at virus and nasal swabs, then traditionally what we have seen in our studies, and I think that is indicative of what other people have seen in their non-human primate studies, is that protection in the upper airways is a higher bar and more difficult to achieve than protection in the lower airways. I think that's seen mostly, pretty much universally with all the vaccines to one extent or another. Why that's the case, we actually are not sure. But here we show very robust virus replication in nasal swabs in the sham control animals. Again, 20 out of 20 sham controls show robust virus replication in the upper respiratory tract. Virus tends to peak on day 2 with median subgenomic RNA copies of about 5.5 logs and between about 3.5 and about 7 logs of virus. Virus replication then persists for approximately 7 to 14 days in the sham controls, very similar to what we saw in our early pathogenesis studies. In the SPP vaccinated group, 5 out of 6 animals showed no virus in nasal swabs, and 1 animal showed a low degree of virus replication in nasal swabs, 2 time points on day 1 and day 2 that were positive. If we look at virus and nasal swabs in the other 6 vaccinated groups, then we saw a degree of partial protection in most of these groups. Between 1 and 3 animals in each group showed breakthrough virus, and in general, the breakthrough virus was actually, I would call it slightly lower in magnitude and slightly shorter in duration than in the sham control groups. But you can see here that the breakthrough virus to nasal swabs is more similar to the controls than the breakthrough virus in BAL was to those controls. If we look at the summary of protective efficacy for all 7 of these vaccines compared with the sham controls, then here we show the bronchoalveolar lavage virus on the left and the nasal swab viral loads on the right. The median peak subgenomic RNA copies in bronchoalveolar lavage was about 5 logs, and in nasal swabs was about 5.5 logs. So fairly robust virus replication in the control group. In the vaccine groups, there was a degree of partial protection in with most of the vaccines, with complete protection in BAL and near complete protection in nasal swabs with the SPP vaccine shown on the end. And also you can see visually that in for the vaccines that were intermediate, then the breakthroughs in nasal swabs was generally more robust than in BAL. Because of the size of the study, being 52 animals, as well as the variation in outcome in terms of some vaccines giving near complete protection and other vaccines giving substantial breakthroughs, we were able to do an immune correlates analysis and actually many of the antibody functions correlated with protection, largely because many of the antibody functions correlated with each other. But the best correlations were seen with the pseudovirus neutralizing antibody titer, here shown at week 2 and even better at week 4, that the log of the pseudovirus neutralization titer inversely correlated with the log of peak subgenomic RNA copies per ml here in bronchoalveolar lavage. In other words, the neutralizing antibody titer induced by the vaccine correlated with protection. The neutralizing antibody titer shown here also correlated with protection in nasal swabs. And together with Galit-Alter, we also did another immune correlates analysis in which we divided all of the vaccinated animals into those that achieved complete protection, which was defined as no virus detectable in BAL or nasal swabs at any time point, and those animals that showed partial protection, which was some virus detected in BAL and or nasal swabs at least one time point. And if we divide the animals into these two groups, then the most significant discriminating parameter was the neutralizing antibody titer shown on the left. In second place was the antibody-dependent NK cell activation response, and in third place was the antibody-dependent cellular phagocytosis response shown on the right. So these data, I would say, suggest, but don't prove, but they do suggest that the neutralizing antibody titer is probably the predominant mode of protection, but that other antibody functions also might correlate and might contribute. And finally, we looked at anamnestic immune responses in these animals. Here we look at neutralizing antibody titers following challenge. Day 0 is the day of challenge, and day 14 is 2 weeks following challenge when the study was terminated. In the sham control group, all of the sham controls developed neutralizing antibody titers by day 14 with titers of about 100, very similar to what we saw in our early pathogenesis studies. In the SPP vaccinated animals, there was really no evidence of an anamnestic response, no increased response from day 0 to day 14. The absence of an anamnestic response suggests that the degree of protection was substantial and that the extent of virus replication in the SPP vaccinated animals was minimal, if any at all. I'm not using the word sterilizing protection because I don't actually know what that word means. I think it's a very vague concept, but certainly the degree of protection in this group was substantial. In contrast, we actually did see anamnestic antibody responses induced in essentially all of the other vaccinated groups. There was one animal in the TPA SPP group shown here and 2 animals in the SDTM PP group shown here that also did not show anamnestic responses, and these animals actually also showed no viral loads and the highest neutralizing titers in those groups. Apart from those animals, which I would emphasize are also included in the 2 groups that received that had immunogens with the proline stabilizing mutations, all of the other animals had clear increases in neutralizing titer by 1/2 log to 1.5 logs, suggesting that if vaccines give suboptimal neutralizing titers such that protection is not complete, then a rapid anamnestic response may contribute to more rapid control of virus compared with what would be seen in a naive animal. And finally, we looked at T-cell responses in these animals post-challenge. These are CD4 T-cell responses shown on the top, and CD8 T-cell responses shown on the top, and CD4 T-cell responses shown on the bottom, 2 weeks following challenge in the sham control animals on the left and in the SPP vaccinated animals on the right. In the sham control animals, 2 weeks following challenge, you can see there is high magnitude and broad-based CD8 and CD4 T-cell responses against multiple viral immunogens, viral antigens, including spike, S1, S2, nucleocapsid, and NS6, 7, and 8, as you would expect from a robust virus infection. In contrast, in the SPP vaccinated animals, 2 weeks following challenge, there was only minimal residual CD8 and CD4 T-cell responses against the spike protein, which was the vaccine antigen, and there was no evidence of induction of T-cell responses against non-vaccine antigens, again providing an orthogonal measure of the extent of protection in these animals. So briefly to conclude, the Ad26 SPP vaccination resulted in complete protection in the lung and near complete protection in the upper respiratory tract. The lack of anamnestic responses, both humoral and cellular, suggests minimal to no virus replication in these animals following a challenge. This study involved a single-shot vaccine, And we believe that a single shot vaccine actually would have major practical and logistic advantages over a two-shot vaccine for mass immunization plans and pandemic control. However, based on our studies with this platform for HIV and Zika, we also expect that a second shot of a homologous Ad26 SPP vaccine would likely increase neutralizing antibody titers by more than tenfold, and early data is suggestive of that. So we actually feel that both a single-shot vaccine regimen as well as a two-shot vaccine regimen with this vaccine are worthy of exploration in humans. We also observed that neutralizing antibody titers appear to be the strongest correlate of protective efficacy, consistent with the prior DNA vaccine study. And if we actually include both the DNA vaccine study and the Ad26 study together, then this correlate has now held up in 87 animals, which starts to be non-trivial numbers. In terms of the development plans for the Ad26 SPP vaccine, so first, it's now called Ad26-CoV2S. The phase 1/2 studies are currently in progress and data is being reviewed. And depending on the outcome of that review, a phase 3 efficacy trial is planned to start over the next week or two. I'll also stop with a couple broad perspectives for vaccines. Vaccine development for COVID-19 is proceeding faster than for any pathogen in history. Of course, that has to be safe and it has to maintain the public trust. Multiple vaccines should be pursued in parallel since it is not clear which will be the most safe, the most protective, the most manufacturable, and the most deployable. We believe that preclinical challenge models may provide useful insights into vaccine potency, efficacy, and immune correlates. And finally, we have to realize that there are 7 billion people in the world, and to achieve herd immunity, a large fraction of these people need to be vaccinated. And so therefore, we need multiple vaccines to be successful. So we don't view that this is a race or a competition amongst different vaccine developers. We actually are hoping that many or even all of the vaccines in advanced development will be successful, so we can hopefully have tools to fight this pandemic. These are the acknowledgements for the Ad26 study in monkeys. My group at Beth Israel Deaconess Medical Center, our industry partner at J&J, as well as a large number of collaborators at Bioqual, University of North Carolina, the Children's Hospital, Reagan Institute, and MIT. And this is a picture of our group last fall and happier times when we could take pictures like this, and hopefully we'll be able to take pictures like this again before too long. Thank you. I'll be happy to take any questions.
Okay, thank you, Dan. That was a fantastic talk to introduce the whole vaccine effort and to talk specifically about your Ad26 platform. Got some questions for you. Are hamsters a good surrogate for NHP for safety and efficacy with your vaccine?
Yeah, thank you for the question. We actually published a paper a few weeks ago. I didn't have time to talk about it today in this talk, but we published a paper on the development of the hamster model, pathogenesis, as well as vaccine protection in that model. And essentially, it's an amplified version of the disease process that we see in monkeys. So with a high dose of a virulent challenge stock, then we see consistent weight loss, which is similar to what other people have seen in the field. We even see partial mortality in hamsters, and the extent of the pneumonia is really profound. There's almost complete consolidative pneumonia and very high levels of virus in lungs, upper respiratory tract, and actually moderate levels of virus in virtually all tissues as well. Vaccine protection was still robust. In that virulent challenge model, then the animals did get infected following vaccination, but clinical disease and mortality was prevented.
Okay, the next question is, it's reported that there's a decline in protection post-infection starting at 3 months. Looks like stimulated immunity is not long-term. Did you challenge your animals past this time to see if the vaccine would hold immunity? It's a durability question.
So durability is a critical question for all vaccine programs. I don't think that I haven't seen durability data for any of the vaccines under development. In the published studies, these were not durability studies. These animals were challenged at 6 weeks. We currently have studies ongoing in monkeys in which we've now kept the animals now up to 10 weeks, and we will keep them for 6 months or even maybe a year or 2 years. Depending on what we can do. So we are essentially currently doing the durability studies. In addition, in the phase 1/2 clinical trials that are ongoing, those individuals will be followed for a period of 2 years. So we will generate 1 to 2 year durability data in both macaques and humans, but those data are not available yet.
Okay, next questions. What about pre-existing immunity against the Ad26 vector?
So in the Western world, it's very rare. In the developing world, such as in sub-Saharan Africa, a majority of individuals do have low titers of baseline Ad26 antibodies. However, we don't believe that those are impactful because in the HIV vaccine program and in the Ebola vaccine program, now with about 100,000 individuals enrolled, then there is no correlation between the low levels of baseline Ad26 antibodies and suppression of immune responses. So what we think is relevant is not the seroprevalence of baseline vector antibodies, but rather the titer of baseline vector antibodies. And the titers that are found in the developing world are almost universally low titers that do not appear to suppress the immunogenicity of this vaccine. A second shot of the vaccine is also very successful. However, after 2 shots of the vaccine, then there are high vector titers that do wane over time. But if you need to give 3 or 4 shots of this vaccine in a short period of time, then there probably would be suppression for the 3rd shot and beyond if they have to be given in a short period of time.
Thanks. In the hamster animal studies, do you see new adaptive mutations in the virus?
So we have fully sequenced our challenge stock, and it matches exactly the original Washington strain. And the issues of challenge stocks is really coming to the forefront because it's clear that different groups are using different challenge stocks with different levels of intrinsic infectivity and potency. We did not sequence virus that came out of the hamsters. We have those samples, so we could do that, but we haven't sequenced the virus from hamsters or from monkeys. We could certainly do that if there's a reason to.